All right, well, let's get started then. If you guys know of other people trying to get in, maybe you can text them or whatever. I put it on the Carmen. I know some people don't have access to the Carmen for whatever reason. I put in all their names and their dot numbers. I'm not sure why it doesn't work. I'm not sure why any of this stuff doesn't work right. But we'll continue on, and I'll have to figure out what to do for Thursday. If you just have to create a new link every time, which would be totally annoying. You guys are in other classes, right? Do other people have this problem? Probably not. Okay. All right, so today's lecture, we get away from the really easy stuff and we're going to get into some concepts I think we first have to understand. Before you can understand anything else about neuroimmunology, it's about immune privilege, blood-brain barrier, and CNS lymphatic system, which are really key concepts that we need to know. So anybody have anything they want to bring up from last time? I feel pretty good about the intro so far. Okay. And again, people use a verbal thing because like with my slides and stuff, I can't really see people nodding or raising their hand or anything. So just unmute yourself and just ask a question. So we're going to talk about immune privilege. We want to talk about the structure and function of the blood-brain barrier. We're going to talk about the circumventricular organs and highlight the emerging concept of CNS lymphatic system. So I think we need to know a little bit of neuroanatomy. And again, this is a sagittal section of the brain, and it highlights two areas, the olfactory bulb and also the right there at the skull here. Let's see if I can make this thing a laser pointer. So this here shows this insert, and you see the parenchyma. Everyone know what that means? Anyone not know what that means? Okay, then I won't tell you what it is if you know what it is.
I should. And as soon as I Google it, I'll be like, yes.
So it's basically a word for like the tissue, the meat. So this would be the white matter, gray matter of the brain, right? And so this is the skull, and then you have the CSF that drains into the sinuses. And then we have the olfactory bulb, which also has drainage into this lymphatics. And so it's pretty well known that this was here. And we'll talk a little bit more about some of these areas. But let's see. So this is another slide of looking at the neuroanatomy. And here on this slide, the skull would be up here. And right below the skull is the dura mater and the arachnoid. And then you have the arachnoid space. So the arachnoid space are filled with these trabeculae, okay? And this is where all your major blood vessels are. I mean, you know that the brain uses, more oxygen than any other tissue. So it has to be, highly innervated by blood vessels. So you see that here. This is where you see the major ones. And then you have the capillaries that branch off and go into the parenchyma. And those little triangle guys that you see on the screen are neuronal cell bodies. Okay, beautiful drawing by me showing you the axon in the cell body. So this is where the major blood vessels are, and then this would be sort of the cortical tissue, and you see the capillaries branch out. So what is immune privilege? So this comes up with something that someone had asked about on last Thursday, I believe, about immune privilege and tissue rejection. So one of the things they found out about the brain was that you could transplant tissue in there without it being rejected. So the idea was that prolonged survival of tissue grafts meant that there is no immune system or lack of immune system or deficient immune system, and that the presence of this blood-brain barrier and lack of lymphatic drainage contribute to this. And very few leukocytes are present in the brain and very low detectable levels of MHC, which is the antigen presentation molecules of the immune system. So the prevailing theory was that there was a passive immunological non-reactivity from this anatomical isolation. So like the blood-brain barrier is basically this bridge or this barrier to a bridge, and you couldn't get past it. But this, as we'll see, immune privilege has changed quite a bit over the last 50 years, particularly in the last 20 years, and even more so in the last 3 years. So it appears that immune regulation is dynamic and actively regulated. We know that lymphocytes move in and out of the CNS, and we'll talk about that. We know more about the resident innate immune cells, the microglia, and we'll talk about them exhaustively probably. And we know that MHC, which is important for antigen presentation, is present in the brain and is inducible. So you can see evidence of antigen presentation within the brain and by cells of the brain, particularly microglia and CNS macrophages. We know that if you inject proteins in the blood in the brain, it'll drain in lymphoid tissue. This will be in the deep cervical lymph nodes. We know that there is definitely a efferent arm of the immune system. Peter Medawar in 1948 demonstrated this. And then Matsuzaki and Perry confirmed that this immune surveillance using a bacteria. And then recently we've seen these lymphatic drainage pathways discovered. So this is that classic experiment from 1948. And this is showing you the first picture is a rabbit, and the little white dot there is a tissue graft. Okay, so they actually were doing skin grafts from other rabbits that were put in there. And so you could put a skin graft into the brain of another rabbit and it was tolerated. Okay, so this is the idea that, you'd say immune privilege, there's no immune response. And in order to get graft rejection, you need an immune response. Okay, so what they did then is they implanted that skin graft into the skin and they found that it was rejected. So if they did that first and then put it in the brain, they found that both grafts were rejected. So the major conclusion, and it's here in the black, is that the skin homografts transported to brain submit to but cannot elicit an immune state. So this was a really classic finding saying that you could have an immune response. And this is sort of the key points I want you to think about as we talk more about immune privilege. And these will absolutely be questions that I will want you to know. And you'll see here that's not on your PDF file, but will be in my lectures on my gold trim around these. And so there'll be a question or two from
the slides itself still have to actually be. Oh God.
Are you on? Is that a question?
No.
Okay. Did you have a comment? No. So the reason I do this is that so you have to, if you want some of these gold trims, that you have to either listen to me live, maybe as dreadful as that is for some of you, but or listen to recording. So the thing about immune privilege is that it's relative and dynamic, and it's really confined to the CNS parenchyma, which is why I was asking if you guys know what that definition is, which you'll need to know for this. Privilege is in the CNS is relative to other organs. So think about that skin example I gave you before. So if you have a skin wound and you scrape and you have tissue remodeling and damage associated with inflammation, oxidative stress, that you regenerate those skin cells. You may have a scar, but you can tolerate that change. So privilege within the brain immune privilege is definitely relative to other organs. It's applicable to both adaptive and innate immunity. It's not solely dependent on the BBB. Okay, so the blood-brain barrier isn't just this wall that you can't get through, and we'll talk more about some of these things. And many areas of the brain are not immune privileged. So ventricles, circumventricular organs, meninges have plenty of immune cells and have circulating immune cells that go through them. So we also know that the CNS has its own specialized resident innate immune cells, the microglia. So again, it's dynamic, dynamic and relative to other organs. So here's a picture from one of your, it's an older review, but really nice. And this really hasn't changed in all the years in terms of what is immune privilege and what is not. And what this slide shows you is how it's compartmentalized. This is a coronal section of the brain. You see the parenchyma, the white matter and the gray matter, which are considered to have immune privilege. There's really no immune cells besides microglia in here. But areas like the choroid plexus, which is the area that makes CSF, the areas, the meninges are not considered immune privileged. The CSF and the ventricles not considered immune privileged. The subcortical organs, which are circumventricular organs, are not considered immune privileged. So a lot of areas are not considered immune privileged depending on where you are. So what is the blood-brain barrier? I think a concept that you have to understand about immune privilege is this blood-brain barrier. And so looking at that slide there, we know that the brain has a lot of vessels, right? Again, I hinted at the brain uses more oxygen and energy than any other tissue. So Paul Endrich here was did a bunch of studies of labeling vessels. And so when you injected dyes into the blood vessels, you see that you'd see the color dyes all throughout the body, but not in the brain. And the dye was a hydrophilic molecule, and found that it was blocked. So there's something different about it. So can any of you guys out there tell me in this slide, where is the blood-brain barrier?
It would be encapsulated temperature.
Exactly, right. So I think that the humorous part about some of this stuff with the blood-brain barrier is when I teach in the medical school and they have the medical immunology textbook, they have what looks like like a square that blocks the entrance of things like somewhere in the neck, right? So, some sort of filter, but that's not the way it is, right? So that the major blood vessels are sort of encapsulated by this blood-brain barrier. And that's sort of the trick of it. And over the years, since these studies with the dyes were in 1885 all the way through 1900s, 1913, they started observing different structures, the microcapillaries, the phagocytosis in the perivascular space, and then identify these tight junctions and metabolic pumps. Okay. And one thing I can tell you is that there is also a spinal cord blood barrier, and it's different than what happens in the brain. The spinal cord blood barrier is older, more ancient, and you see sort of less structure integrity, I think, especially in inflammatory responses. You see that a lot of times within the brain, these are immune cells, these are neutrophils coming into an area of injury. And you find that the brain oftentimes confines things to the VRS or the perivascular space, and the spinal cord that doesn't happen. This is kind of showing you that. We'll get into this more, but this layer here, this is the brain side. This is what's called the glial limitin. This is actually one of the major deterrents for things getting into the brain. This is the endothelial cells. These little purple bars are the tight junctions. For an immune cell to get into the brain, it has to pass actually in between the cells, and it usually gets stuck in this Vasco-Robbins space or perivascular space, which we'll talk about. Okay, so the blood-brain barrier, the brain and spinal cord represent 2% of the human body weight, but require 20% of the oxygen, and the brain uses 60% of the body's glucose energy. And the brain has a number of GLUT1 transporters, which are the non-insulin sensitive ones, means they can always bring in glucose. So your brain takes in a lot of glucose. This blood-brain barrier is a physical barrier because of all these tight junctions, those purple little boxes you saw on the slide before. And it forces most cellular traffic to move in between the cells. And the blood-brain barrier protects the brain because the CSF provides osmotic and pH balance, optimal for neural transmission, this very low protein concentration. So that's really one of the keys. And we'll talk about this. There's two fluids that's there, the interstitial fluid and the CSF. And it turns out that having high protein, high iron, and a lot of cells in the sort of buffer system of the brain is very bad. So there's a whole reason why we have the brain is bathed in in interstitial fluid and CSF as opposed to blood. We know that astrocytes and microglia are intimately associated with the blood-brain barrier, and we'll see that, and the astrocyte end feet actually make that glial limited. Now, the blood-brain barrier also is a key area of immune to brain communication, and we'll see some evidence of that as well. So the blood-brain barrier endothelial cells will have transporters and receptors that can respond to signals in the blood and then transmit that signal to the brain. So this is really key, I think, when we think about neuroimmunology, we think about how the immune system and brain communicate, and then endothelial cells of the blood-brain barrier become, really important for that. So I'd like you to know the key concepts on this slide as well. This is more of the structure of the blood-brain barrier, and you can see it. I'm not sure who answered that question. I'm sorry, I just heard your voice, but you're absolutely right that what you have here is a blood vessel, and you have, the endothelial cells wrapping that blood vessels with these tight junctions here, and then you have these astrocytic end feet. Parasites are these little muscle cells here that surround, and then you have neurons. So you have this sort of what's called the neurovascular unit in this area here. And so it basically holds things out. So you see this blood-brain barrier, the tight junctions, and then this what's called the glial limitin here, which is formed by the astrocytic end feet. And then you see microglia, right behind it. So it turns out that big peripheral immune cells don't easily cross this barrier. Inflammatory molecules, these hydrophilic molecules do not cross. And it's important that we maintain the integrity of the CSF and interstitial fluid. Because if you think about it, they're the ultimate buffer for neurotransmission, right? So if you interfere with that, you're going to affect neurohomeostasis, right? So this is also kind of my schematic that I hope will help you visualize what's happening here in terms of this, peripheral blood and these endothelial cells wrapping these vessels with the tight junctions. And then on the brain side, you see astrocytes and microglia lining this, and you see that there's no blood in here. So they're able to take the nutrients of the blood, they're able to take the sugar, the glucose, but they're not taking all the blood proteins and all the cellularity of the blood. So we're keeping that pure, if you will, and we're allowing that to stay. And so it's really important to keep this separation between the blood and the brain extracellular fluid and the cerebrospinal fluid. Okay. Does anybody have any questions about this so far? I know there's a lot here, and maybe it's not as straightforward.
Where are the neurons in this diagram, or are not they like in contact with the blood-brain barrier?
Where?
Yes.
Probably within that astrocyte intersection there, within sort of by the end feet.
Okay.
But they're not, you kind of keep the neurons away from this barrier to some degree. They're part of that neurovascular unit, but you don't see a lot of neurons in that spot, mostly because a lot of things that you're trying to protect the neurons from, is that isolation from those blood proteins and the cells in the blood and the high iron content.
Thanks.
Do we know what the function of neurons is, like in conjunction with like reading the blood-brain barrier? Are they like detecting certain molecules in the blood, or?
They aren't directly, but indirectly, you do have that sensing. You have that sensing from astrocytes. You have that sensing from endothelial cells, and certainly those signals are transmitted across. And we'll talk about how that happens. Probably not today, though.
So are astrocytes giving the nutrients to the neurons, or like how are the neurons even getting nutrients?
Yep. Well, the nutrients are in the fluid, so you will see it get absorbed. There's a ton of transporters on those endothelial cells that transport things across, right? But astrocytes are very important support cells for neurons. So you're right there. They do help with that metabolism.
You called pericytes little muscle cells. What exactly does that mean?
So any vessel will have some muscle cell. So a pericyte is a muscle cell, and it helps, I think, give the contractivity of those vessels. It's common. All vessels have some parasites.
Do you know if the parasites around the blood-brain barrier are the same as those in the periphery?
I think they are likely specialized within the CNS tissue. Any cell within the brain or spinal cord has some specialization. So I would say if you were to do single cell sequencing between those of the periphery versus the CNS, you'd find some differences. Okay. Well, thanks a lot for the questions. Glad you guys are paying attention. So I'm going to add one more layer of complication for you, okay, is that the blood-brain barrier isn't everywhere in the brain, okay? So there are some access points. And this is where circumventricular organs come into play. So structurally, this is a rat brain. So in case you've gotten used to looking at some of my sagittal or coronal human brain sections, this is from a rat, okay? So this is a rodent brain. And what you can see here, the gray, or sorry, white parts here are the brain parenchyma, okay? This light gray are the ventricles. So this is where that CSF is, okay? So you have CSF within here. And the subcortical organs or circumventricular organs are areas that surround the ventricles, right? Circumventricular organs. So they're a little specialized area within the brain that surround the ventricles. And so here's a whole list of them. I'm not going to ask you to know these. You might see them in some papers and things. I can just list them for you, but I would never ask you which areas are circumventricular organs. It's not a neuroanatomy class, but you'll see things like the medial eminence, the pineal gland, the subfornical organ, the area postrema, the lateral terminalis, and you can see them here within the ventricles. Anybody know why the ventricles are there? Any neuroscience grad students out there?
For the circulation of CSF,
Yeah,
for
but
the
why?
production of it.
Why is that important?
To drain stuff, just like how the lymphoid tissue, lymph nodes work in the periphery.
Yeah, I mean, it's sort of like if you think about when they used to build mills along rivers, and they would, dump stuff in the river, or they'd send stuff down the river to the next one. They'd send the logs down the river to the mill. So it's a transport system, both to and from. So it allows you. So if you ever want to get something in the brain and for it to go everywhere, let's say you have a virus, you have a free virus, and you want recombination throughout the brain, you just put it in the ventricles, right? It's gonna go everywhere. So it's like that same idea. Okay, so here's my picture again of the blood-brain barrier. You'll see it's aligned again with the astrocytes and the microglia. This again is called the glial limitin, right? But there are areas within the brain that don't have the same structural integrity of these endothelial cells. And one of these areas, or these areas in general, are the circumventricular organs. So they still have astrocytes, they still have microglia, and they still have this sort of glial limitin layer. So you're not getting blood flow in there. But in terms of neuroimmunology, you have a lot of sampling of what's happening in the blood. So again, it's not neurons that are sampling, it's going to be the glia, the astrocytes, and the microglia that are involved of this sampling. So there's a lot of things that happen within the CVOs because they lack, a reliable integrity structure of the blood-brain barrier. It's still there. It's just there's still endothelial cells there. It's just not the same tight junctions. So I think in terms of neuroimmunology, the reason this slide again is highlighted is that I want you to know these are areas of immune surveillance. There are, we see them along these midline structures bordering the third and fourth ventricle. These are not considered immune privileged. They are outside the blood-brain barrier. They also see, so if you were to sample these areas, so within the CSF, you do find cycling T and B cells, which are, cells of the peripheral immune system. And, we can see these circumventricular organs as key areas of immune to brain signaling, the way the immune system can signal the brain. Okay, so we're going to get into a little bit more of this concept of the cerebrospinal fluid and the interstitial fluid and how they drain the CNS. This is a picture from an immunology textbook showing you the lymphatic system within the periphery. We know that it drains everywhere. That basically it uses the lymph drains based on muscle contraction, right? And it drains to the lymph nodes where we have immune cells. And we see that, when we have an antigen within the lymph node, we know that it gets picked up and we'll have an immune response, right? So the brain doesn't have the same structure, but it has some important factors. Okay, so I do want you to know that there are two fluids within the brain and spinal cord. They're the extrastitial fluids, the cerebrospinal fluid, and the interstitial fluid. So the CSF comes from the choroid plexus. So the choroid plexus is a pump, and you'll see in some of the upcoming slides is that the choroid plexus pumps CSF in a direction. So it pushes it out. From the choroid plexus, it goes into the lateral, and then the third and fourth ventricle. It flows from the ventricles to the subarachnoid space. So that's actually getting kind of out of the brain, and all of it will drain. So the brain parenchyma, so the meat of the tissue, the white and gray matter, is bathed in the interstitial fluid. Cerebral endothelial cells make interstitial fluid, and it's derived from blood. So there's a lot of components of the interstitial fluid that look like blood. It's just the blood without the cells, the iron, the red blood cells. So it's made by cerebral endothelial cells, and that's a lot of how the nutrients are transferred. So there's some overlap. About 15% of ISF drains or into CSF, but they drain by distinct and separate pathways. So the CSF, you can imagine, provides the brain and spinal cord with sort of a fluid pad, if you will. All right, so if you don't have CSF, your brain would sink or would be knocking against the skull. It also allows a transport route by which neurotransmitters, hormones, cytokines travel to wider areas within the CNS. And also, as one of the students mentioned, it's a way that waste is removed. So the common idea is that the CNS did not have a lymph drainage system like the periphery does. But remember that CSF and interstitial fluid are drained. All right, so they're drained. In humans, a lot of it actually drains directly into the blood. And I think in humans, within a few hours, or let's see, I have it down here. We'll talk about that. So CSF is secreted by the choroid plexus. Like I said, it goes through the ventricles, it's pushed through there, and then it flows through the subarachnoid space. It uses connective movements to do this. So if I was to inject tracers into the ventricles, we'll see two major routes. We'll find that the CSF will drain into the olfactory bulbs, through the nasal lymphatics to the cervical lymph nodes. Okay, so you do see drainage of the CSF, and then also can drain through the subarachnoid space directly into the blood. So in rodents, 50% of CSF drains to the deep cervical lymph nodes, and 50% of it drains through the subarachnoid space, meaning that there is a recycling of CSF, and it is dumped into the bloodstream, or it does drain into lymph nodes. So this is not really the mystery of the lymphatic system. We'll explain more about that. So humans, most of it drains directly into the blood. A small amount of it drains into the nasal mucosa and nasal lymphatics. CSF drainage in man is quite a lot. It's like 520 mils per day. So you're recycling all your CSF within a few days, and the rat, it drains about 4 mils per day. And this is the major routes of the way drainage goes. And so this would be the top view. Choroid plexus here is pumping out the CSF, and it flows through, and you see it flowing through the ventricles. and then it sort of flows out either to the subarachnoid space or in through these perivascular spaces into the drains through the into the deep cervical lymph nodes through the nasal lymph system. So rodents, about 50% goes through this olfactory bulb, nasal lymph to deep cervical lymph nodes. In humans, it's about 10%. In humans, most of the CSF drains through the subarachnoid space, through the arachnoid villi to the venous blood. So in humans, most of it dumps directly into the blood. So you can imagine if there's something within your brain, right, and it drains through the CSF, it will go into the bloodstream. And I think these come from some of the papers that I put into the Carmen account, the Weller 2009. Okay, so.
Dr. Godbout, I just have a question if you don't mind.
Sure, absolutely.
I might have missed this, but is this because of the different anatomy of human versus rodents, or is it because maybe something like a fat soluble versus water soluble, like differences between human and rat CSF?
I think it's a evolutionary structural change. I think it's the higher order mammals drain more directly into the bloodstream. So I think it's, for whatever reason, it's an improvement in biology. I'm not exactly sure why. I'm not an expert on CSF drainage, but I know that it is, as you move up the higher order mammals, it's more drainage directly into the blood.
Sure, thank you.
Okay, so the cerebral endothelial cells make the interstitial fluid. It's supported by mitochondria, which is the energy of the cell. These polarized sodium and potassium pumps. So the energy movement from the CNS is dependent on the production and continuous flow of interstitial fluid. So it drives solutes in the opposite direction of the blood flow. Interstitial fluid is sort of the optimum media or buffer for neuronal function. Similar composition to blood, but its solute content and cellularity are significantly lower. So again, for the main difference between ISF and blood is, there's no cells, there's no iron, and it's low in protein, whereas blood's high in protein, and there's no cells. That's why it looks clear versus blood, And I don't know if you guys have ever, watched any medical shows in my day, which I guess dates me a long time, is ER. And we used to watch that in college, and they're always checking the spinal tap, right, to see if it was clear. If it wasn't clear, something bad was happening, right? So you generally do not want blood or cells within the CSF because it's really bad for neurotransmission. You can inject things into the blood, into the brain, tracers into the parenchyma, and you'll see that it drains. And they've done this. They've been doing drainage studies since the 40s. We see that it moves along the basement membranes, and we see that it sort of passes over perivascular spaces. And this becomes important for neuroimmunology because, again, if you're going to have this flow of interstitial fluid and it's flowing over perivascular spaces where you have this concentration of macrophages, we're going to see that it drains out the base of the skull to the cervical lymph nodes. Now, cells can't leave this using this route, all right? So an immune cell can't travel out through these subarachnoid spaces. This is just showing you the movement of interstitial fluid. And so this is that same diagram I showed you before with the skull would be up here. You have the dura mater underneath it, and that subarachnoid space where you have these big vessels, right? So the vessels are bringing blood, the capillaries come through the parenchyma, and that the interstitial fluid actually moves against the direction of the blood. And you're seeing it moved through the basement membrane. It's passing over these perivascular spaces, the space between endothelial cells and the parenchyma. and it can pick things up, debris and things, and pass out and goes out through the base of the skull. Okay, so in big cap letters here, this is where that lymphatic system comes into play, because immune cells do not drain out this fashion. So while those arachnoid villi allow CSF to flow through to the subarachnoid space and then leave into the bloodstream, you can get water and small compounds in the bloodstream, but larger compounds, especially immune cells, are not transferred through this route. So this is the difference here. This is the difference in, why we have this newly identified lymphatic system. So this is one of the guys that found this, or really brought this to the forefront, was Jonathan Kipnis, who was at UVA at the time, now is at WashU. But he had a sort of one of the landmark papers to show this lymphatic system, and basically using labeling for vessels that are normally found in the peripheral lymphatic system, found that it was located in the meninges within the dura mater, enables fluid, macromolecules, immune cells to drain from the CNS into deep cervical lymph nodes. So to answer, I'm sorry, again, when you guys ask questions, I can't see who it is. But this is part of the point too, I think, is that we don't need the drainage, that same drainage system that the rodents have, because we have this more advanced lymphatic system. These are the two papers that came out a few years ago. Antoine Laveau, who's now at the Cleveland Clinic, was the first author of the Nature Letter paper. And then there's Asplund and Alito here from Journal of Experimental Medicine. And both of them had really similar studies showing this lymphatic system. And this is some work from these papers showing you, basically all they're doing is labeling for lymph tissue within the brain, and they're looking within the meninges. And so the reason that people really didn't see this before is that, how many of you guys have done brain dissections, right? And I think some of you just may have done some this morning. So you pull the brain out of the skull, and you scrape that little tissue. There's a little kind of a white tissue that comes off as you're kind of pulling it away from the skull. That's the meninges. So most people in dissections do not take out the meninges. So if you can isolate the meninges or not destroy it when you're doing your sectioning, you can start to see that these lymphatic system are within the meninges. Okay, so this is a paper here showing you, this is sort of the neuroanatomy. I think Kipnis draws it a little nicer than I've been drawing it for you, seeing sort of the flow of CSF and interstitial fluid. This is the blood capillary coming in. There's nutrient exchange here in these capillary beds. You see that blood-brain barrier. You see these are microglia, these are astrocytes, these are neurons. You see this exchange. And then you have these post-capillary venues. Now you have this. So where the lymphatic system is within the dura, so within this meningeal lymphatic system. So the CSF will drain here. Okay, so this will allow cells to drain out it as well. So it's another drainage system. And this is showing you this, revisiting the mechanism of immune privilege, showing you the two types of drainage. Again, so this would be the common CSF draining through the sinuses, whereas now with this lymphatic system in green, you see that within the meninges, we can have drainage of immune cells, pathogens, and other things that drain directly to the lymph nodes. All right, so this is a big step forward in showing that we do have a lymphatic system within the brain, and it's within the meninges. Just showing you more of the neuroanatomy of it. So you're going to have, again, you see it here in the dura mater. You see the green lines here. It kind of looks like to me like an airport a little bit, like the landing strips and the little. Anyway, bad joke. But yeah, the drainage is here in green. This is the you're going to see the flow of the CSF through the subarachnoid space here. And we're going to have, immune cells and other macromolecules drain through here. So back to the diagram I showed you guys before, with adding the lymphatic system on it, where is it? You can see here it's within the dura. And as I said, oftentimes when you pull off the skull in a dissection, you pull off the meninges and the dura matter as well. So if you were ever, looking for this before, you didn't have intact meninges, you'd never find it. And that's really the advancement, I think, that actually Laveau had really was the guy that did this technique and was able to isolate the meninges. Now, I mean, they isolate cells from the meninges and they capture things within this vessel and do like cell sequencing on them. So they've gotten pretty advanced in their ability to capture those cells. So this slide is in yellow. So you can, I want you to know where the lymphatic system is compared to, the drainage of the CSF. And this, I think, is my last slide. Let's see. Oh, no, second to last slide, I think. So one of the ideas too, and for those of you thinking about a topic for your paper, it's a hot topic. And the idea is that we have this normal drainage. And we have the drainage of the CSF, and that are some of these immune cells that cycle through and other things that get sensed by the immune system drain through this lymphatic system in the meninges, right? So it's part of our normal drainage. It's way that we can get things to the lymph nodes and for the immune system to respond if needed. And that with age or disease, if we have dysfunctional meningeal lymphatic system, so one of the ideas with Alzheimer's is that you have this buildup of debris and it's not being cleared. We're seeing this reduced drainage, and this is where I think people are very interested, in understanding the system and how it changes with age or disease. So this would be an interesting topic to explore. Okay, so the summary of the lymphatic system is that the CSF drains into the bloodstream, but these macromolecules and immune cells will drain through these meningeal lymphatic vessels, and they'll drain to deep cervical lymph nodes. So if we were to inject something into that dura and get it into the glial lymphatics, we'll see it drain to deep cervical lymph nodes. So cervical lymph nodes are all the ones that are in your neck. So you'll see drainage within there. I think I just said this, but if you do the tracing with the macromolecules, you'll see that they go drain through these ways. And other people have actually shown this drainage path. I think, what Kipnis and the other guy did, there was actually a paper in the 90s that seemed to identify the lymphatic system as well, but nobody believed it. And they didn't have the same technology that we have now, but they did see, drainage from the dura into the deep cervical lymph nodes. It just, sometimes science is not the novel idea, but actually having the technology to really show people and convince people. So immune cells, T cells, B cells, and dendritic cells were present in the lymphatic system under normal conditions. So they are involved in this normal trafficking. So we know immune cells get into the brain. We know they cycle through. There's plenty of examples. If you prevent immune cells from surveying the brain, you can get these really rare, seen as viruses. So we know that happens. Now we know how they get in, and we know how they get out. Okay. So this is something also from Kipnis's paper. He says that the endothelial cells of the meningeal lymphatic system may be uniquely equipped to ensure tolerance of T cells reactive to CNS-derived antigens. When the mechanisms of lymphatic endothelial cell immune tolerance fail, autoimmunity may develop. Therefore, classic lymphatic vessels draining of CNS may in fact be fundamental to ensuring CNS privilege. So basically saying that there's some specialties of this drainage system that protect the brain against immunoreactivity. So I think that's all for today, and I think that keeps us on time today. Let's see if I can stop sharing, and I will probably stop recording too.